welcome to What's Now San Francisco. We've been at this now for almost a year, actually. It's been a program that's been, quite successful. We get amazing speakers, and tonight is no different. And we get a really, really great crowd, and it's a very interactive program. So I always like to ask the question, who's here for their first What's Now? Yeah, it's always a good big part of the room. So thank you all for coming for your first time. For those of you who don't know who Capgemini is, we're a global consulting, systems integration, and outsourcing company. We're based in Paris, France. We have about 200,000 people worldwide. We operate in over 40 countries, and we're about a $13 billion company globally. And one of the things we did about a year and a half ago, we spun up a practice area that we refer to as applied innovation. And as part of that practice area, we've established 10 applied innovation exchanges around the world. We just opened one in Singapore a couple of months ago. We'll be opening one in New York this summer. And this is our flagship facility right here in San Francisco. We've been here a little over a year. And one of the things we did when we opened this, we engaged with Peter Leiden from Reinvent, and we strategized over what is something we could do here that was a little bit unique and a way for us to, as kind of the new kids on the block, even though we're big kids perhaps, it was kind of our first foray into the Bay Area, and what could we do to really engage the community around innovation. And Peter and his team and their infinite wisdom worked with us to come up with this concept that Peter will explain in a little more detail when he gets up here. The thing about tonight that I find very, very interesting, we've done a number of these events. I don't know if we're up to number 8 or number 9. I've kind of lost count now. We're over 10. Say I lost count. I was never taught to count more than 8 or 9, so that's part of the issue. But it's interesting because I think all these events are very profound and very provocative in their own way. But we did one, I think it was 2 events ago, on sustainability with Paul Hawken that, to me, really stood out because it was really about our future, it was about society, it was about the world, and it wasn't just about technology and how it would be applied to business. It was more profound than that. And tonight, I think, is going to be very much the same. We have Andrew Hessel here, fantastic speaker, very prominent. Again, Peter will do the introduction. But again, I think tonight is going to be also of that same vein, where it's going to be very provocative and profound of how lives in the world and our futures can really be affected by some of the innovation going on in a very unique space in the market. So I'm really excited for tonight. So I'm going to turn over to my partner in this program, Pete Leiden, and I want you to pay specific attention when I hand this microphone over to Pete, because I always like to bring in one current event when we do this. And the current event I'm going to bring in is that little hand slap that Donald and Melania did, okay? So look very carefully when this microphone's passed and see if you can pick up on the hand gesture, okay? All right, Peter Leiden.
Okay, here we go. Lanny. Thank you very much. Since there's a lot of new folks here, I will just add one little thing here to what's now. I mean, one of the things, framing and the kind of mindset we come from the beginning here, is that we're living in a really extraordinary time. I mean, the transformation going with digital technologies kind of sweeping through, disrupting all kinds of things, all the transformation in the global, kind of globalization of everything, working on increasingly planetary scale, the beginning of a kind of era of sustainability in a much more aggressive way in the face of climate change, and also the opening up now, as we're going to talk about here, bioscience, all the different spaces in there that are just going through really big changes. And the idea here is that there's really no better place to be on the planet right now than San Francisco, to really see so many of these things are happening here. And it's not to say other places aren't doing awesome things too and having different pieces of the puzzle, but if there's one place on the planet you really want to be today, it'd be San Francisco. And so the kind of device, or the idea of this whole series was to say, hey, let's go to each month, take a different space, that really is just exploding innovation. Get a real remarkable person in that space to explain not only just what's the cutting edge of what's happening in that space, but explain it in a bigger context of how it fits in this larger whole. Now we've done, like I say, as Lanny miscounted there, we're over a year, and this is our second year, and now we're moving past into about a dozen of these things. And one of the things that we haven't done is we haven't really moved into the life science space. And there's a couple of reasons for that. But one of them, because if there's, this is genetics, biotech, neuroscience, life extension, stem cells. I mean, there's tons of things happening. In fact, not far from here, Mission Bay, across the bay and the East Bay as well, are all over the region here. That's just profoundly important things that are going on in development, but we haven't really touched them yet. Now, one of the reasons for that is in my own expertise, and that is more limited. And I didn't feel I'd be a really good guide to actually move us into that space. And so what we've done is we've recruited one of the best in the business here, David Ewing Duncan, who's going to actually take my role today. And because of his background. Now, David has been over 15 to 20 years, has been really a journalist in this space, going deep, making the connections between the really most remarkable players in there, gaining their trust, understanding what they're doing. He's also been understanding the pros and the cons of what's happening in these spaces. He's actually giving it kind of judgment, and he's been able to interpret it to wider audiences. As a journalist, he's also an entrepreneur. He's started his own series, you could say, an event series called Arc Fusion that is a more occasional series, but it also very similar to kind of the gatherings we have here. And so we're really delighted that he is going to basically today, host this one. But I will say more than that. We at Reinvent, the company I founded, and that's driving this here, is we're actually developing a whole series around in this life science space, and that we would love to have. We're developing an idea that each month to go deep into one of these areas in the life sciences space, the bioscience space, and that's one that David's going to help guide us through. It's not up and going yet, but if you're interested, you can talk to myself or David afterwards. But for now, I'm going to hand this over to David, and he's going to introduce our speaker. David. Oh, you don't need it. That's a firm
Hey, well, thank you all for coming. And I have actually known Peter for many years. And we got together, I don't know, I think for drinks nearby here about a year and a half ago, and he told me about this series he was doing. And am I working? Yay, okay. And I gotta say, he told me about this, and I said, what are you doing? But I think what's happening here is something that is near and dear to me as a journalist. I write long-form journalism, which almost died, but it seems to be coming back again. These deep dives into stories, and occasionally I'll do a book or something else. And so this kind of evening, where you take one subject and one person, and you really plunge into it, is so exciting. And I'm really, really pleased, and want to thank Peter for asking me to help out in the healthcare and the biomedical space. I, as a reporter, have covered lots of things over the years. I was a political reporter, foreign correspondent, et cetera, et cetera. But I found out about healthcare and then biotech, and I have stuck with that for almost 20 years, because I think this is where everything comes together. I mean, there's been some amazing programs here. There's obviously lots of things happening in various fields. But to me, the 21st century is all about bringing all of this together. And my little company, Arc Fusion, the fusion idea is the fusion of health, IT, and biomedicine and everything else. So that's what we're going to focus on tonight, and we're going to focus on this if we end up doing this series, which I hope we do. And we're going to get into all those things that Peter was talking about. Neuroscience, stem cells, gene editing, and you're hearing all these things. For those of you all that aren't in the field, you're hearing about these things here and there. And so we're going to try to deep dive into that, but in a way, bring it all together, and try to make us understand some of the bigger pictures. So it's with great pleasure that I introduce our speaker tonight, and the gentleman that's going to be the focus of what we're doing. And when I was talking to Peter, and we were contemplating what to do with the idea of having a longer series, and how do we introduce this? And there are only a few people, and by the way, as a journalist, I am privileged to hang out with people like Andrew. I'm not a scientist, but I get to spend time with. I've been taught by these amazing geniuses, you know, because when I first started writing about genetics, I knew nothing about genetics. And what better way to be taught than by some of the greatest experts in the world who took me aside and said, you know, wait a minute, before you write this, you should probably actually understand a little bit about it. So I know a little bit about it. But Andrew is such an interesting person, as well as being a scientist. He started out, he's Canadian, north of the border. He started out in a fairly traditional way, you know, molecular biologist, geneticist, worked in pharma. And then he had this life pivot where he decided to go out and really try to figure out what was going wrong, what could go right. He's one of those optimistic people, but also realistic about what's happening. And he's gone through a journey, which is extraordinary. And I met him at Singularity University the very first time. Actually, it was almost the first week of Singularity in 2009 or around that time. And I immediately saw a man who, like some people I get to hang out with, lives a little bit in the future. He's thinking about all, you know, how to bring all these things together I was talking about. And he's working in not only serious science, but also how we as a community participate, you know, the open science movement, all of that. But unlike some people who get a little dew-eyed about that, he also is very realistic. And he wrote an Atlantic story, which you all should look up and read. It was in 2012, about, what was the title of it? It was How We Hacking the President. Now, this is taking on a sort of new resonance, perhaps, but it's how you would design a virus that you could infect. Basically, they created a scenario where everybody at Harvard is infected by a virus that gives them a little bit of a flu or a little sniffles, but it's actually genetically targeted for the president who is about to speak at Harvard. And this is just a, you know, kind of sci-fi scenario that they made up, but maybe not so sci-fi. So anyway, so Andrew was very active in Singularity. He still is. He started something called the Pink Army Collective, which was the idea of a cooperative around funding drug development. And more recently, he's been at Autodesk, which is interesting to have a biologist and somebody like him. I think he went there just to kind of say, maybe you should do this, maybe you should try that. You know, 3D printing tissue, 3D printing cells, et cetera. And he's developed that into some really exciting things. But mostly he's lately been working in the overall realm of synthetic biology, which is one of those somewhat nebulous terms, but it basically, the way he's working on it, it's how we humans are going to be and already are evolving organisms ourselves and probably us at some point. So this is really heavy stuff. And this is really where we as a species take off in one direction or another. So I don't want to steal your thunder here, Andrew, so let me welcome Andrew. And he's going to speak for a little bit and show you some amazing things, and then we're going to get up and have a conversation. And then, you know, as these evenings are, we want to welcome you all for most of the rest of the time to join in the conversation. So, Andrew Hessel.
Now, I want to be clear. I don't take anything too seriously. That's part of the joy of doing the work that I do, because exploring the future, you just don't know what's going to happen. I'm just going to dive straight in. Technology has been a big part of my life. It's been a big part of everyone's life here in the Bay Area. But technology is what really allows us to explore, whether we're going down deep with the LHC and trying to look at the smallest things in life, or pushing the boundaries and going off of our planet. It allows us to see in new ways, whether you're looking down a microscope or looking outwards with the Hubble. And I personally really like looking down. This is just a drop of pond water. There are alien creatures in this drop of pond water that will kind of blow us away. And that's because we really didn't have microscopy until just about 400 years ago. Looking up, we always saw the stars. Looking down is really, really new to us. When you move beyond light microscopy, you start needing electron microscopy. Now, these are just some viral capsids under an electron microscope, but it kind of reminds me of the photographs that you would see, from the 1800s. This guy, by the way, Robert Cornelius, is credited with doing the first selfie in 1839. Yeah, if you ever see someone with a selfie stick, blame him. But the resolution is really low with electron microscopy. And that's why we've developed techniques like cryo-EM, which allows us to take objects filmed under electron microscopy, kind of merge the images, computationally analyze. And this is all very similar to what we do with space images. And then be able to look at all the different orientations and then reconstruct it into 3D models that actually have enough resolution that we can start to look at this molecule and rotate it. This is pretty state-of-the-art stuff. And it's opening up the world of molecular biology in ways that we just haven't been able to do before with even our electron microscopes. And of course, all of this today, we start to put into the computer world. This is a modeling software that's actually based on design software that allows us to see all the molecular structures in high resolution, be able to rotate and manipulate these models. This is developed by Autodesk, and it's a big reason why, as a scientist, I joined Autodesk about 5 years ago in a nascent bio-nano group, now a life science group, because you need computational tools to be able to start to explore this world. Now, most people aren't familiar with Autodesk, but we've been around for 35 years. We're best known for AutoCAD, which is used for designing buildings, but we have explorations in things like robotics, we have manufacturing of all different types. If you've ever seen a movie with a special effect, it's our software. We're really big in 3D printing, have been exploring kind of the forefront of manufacturing. In fact, we used to just make software, ones and zeros. Now we're actually learning how to better connect it into the real world. Video games are all made using the types of tools that we use. But the stuff that I really enjoy is we started to explore how you go and design living things as well. And I'm very fortunate that I get to work in a facility not too far from here, just on the Embarcadero, called Pier 9, where we have a kind of the intersection of computer-aided design, computer-aided manufacturing, and some really interesting explorations into the space. And that's where our life science group is based. The image that just played there was one of our researchers in 3D space actually interacting with the molecules. We finally have a tagline that I think is actually pretty good: just make anything. And clearly our software tools are pretty good, but more and more, I know we can pretty much model anything that we have the physics of. Biophysics we're still a little sketchy on, but more and more, our ability to make is actually defined by our printers. So I think a lot about printers, and not just printers for a sheet of paper, not 3D printers, the extremes of these technologies. Here's an example of an extreme. This is the first 3D print ever done in space. Some friends of mine made in space, founded at Singularity University, prototyped and delivered a 3D printer to the space station, and this was the first printer that came off of it, which was pretty cool. It's a magic box to the scientists. Because they don't actually have to design anything. They just say, we want this, and boom, the printer, the black box just makes it. More than that, they've already started thinking about how do we use 3D printing not inside the space station, but outside the space station to build structures that don't have to survive all the stresses of launch, all the G-forces and all the loads of launch. And now we can start to imagine building giant structures just made in situ in zero gravity. This is really a cool area of 3D printing that's just starting to open up. Or, again, we've been making buildings with construction crews. What if you could just 3D print buildings? A friend of mine, Stephen Keating, awesome story in medicine, but also he made and built this printer that can actually print whole buildings extremely quickly. Now in life science, this is what we typically think of as the printer, the person doing the work, making the things. It's someone working at a lab bench. This is far too clean. This is clearly a stock image. But someone working with a liquid handling device, a pipette, all of the reagents. This is a complicated kitchen. Personally, I hate this because if you can see the liquids you are working with, you have 1,000,000 times too much. I'm really interested in biological robots, and that means a lot of liquid handling because we're squishy. Here's an example of that. This is a device that actually uses digital fluidics. It is actually an electronic circuit that can move liquids around, and you can do mixing, you can do incubation, you can do all of the work that a lab tech would do, but now it's all done, highly parallelized, all controlled by computer. It's used for preparing genetic libraries in this case. It's a device you can buy off the shelf. But this is a remarkable way to control fluids. There's other ways, though. This is actually a device that uses sound waves to essentially dispense small amounts of liquid, in this case 2.5 nanoliters at a go, essentially into a reaction well. You can do streams of different droplets. It doesn't matter about the viscosity. The sensors pick up all of this. This is how you do. You use no tips, no consumables. You can now dispense massive amounts of liquids in very controlled ways. And how that's being used is in technologies like this. This is a similar system. It uses inkjet printing, which is essentially a nanofluidic dispensing device, to actually do one of my favorite type of printers, which is the DNA synthesizer. Here in this illustration, they're actually building DNA chains in parallel on a microfluidic system using an inkjet that sprays chemicals. This is fantastic stuff. And it's changing my world. I'll talk more about DNA synthesis in a minute. But you can also use living cells as ink. This is 3D printed tissue. Essentially, it's leather made by a group called Modern Meadow. Now, we're not going to get 3D printers that can do human tissues right away, but people like Andrew Pelling from Canada have been learning how to do things like grow human tissues on things like fruit, using apples in this case as the extracellular matrix to grow and embed new tissues into. 3D tissue printing is one of the most exciting areas of biotechnology because you're actually using living cells as ink. Here was an example that I just pulled out for reference that I thought was pretty awesome. This is a little bit of 3D printed gelatin that they actually inoculate with cells, in this case follicular cells of a mouse ovary. And when they put this little bit of tissue into a mouse that has been sterilized, the ovary is removed, it allows the mouse to ovulate and restores reproductive capacity. Here you see a mouse glowing green. That is actually a mouse that came from this engineered bioprinted ovary that has a green fluorescent protein added to the organism. This is just a honeybee flying. This is one of the things, the area that I think is going to be so much really fascinating is when we start to actually use 3D printed muscle to make robots. Because look at the muscular control over these wings. That's just a few cells. Here's the most recent example I could find of using muscle cells for locomotion. You can see these things kind of creeping along like a little inchworm. Yeah, we got a ways to go. But it's a start. And this is actually electronically controllable. The Hertz signs there, depending on the speed of the electrical input you put in, the faster the robot goes. So it's going to be a while before we have 3D printers like we saw in the movie The Fifth Element. Yeah, I would really like that. Yeah, that's not the way we're going to get rebuilt. And nor are we going to have 3D printers a little more modern with Westworld. But these science fiction stories prime us for what our expectations are in the future. I would love for us to be able to 3D print any tissue, any structure, and biological robots moving forward. It's just all part of the evolution. Autodesk software, which, by the way, made this image, can easily support the design. It's the printers and the cell biology that's limiting us today. Speaking of cells, these are my favorite printers. This is just a time lapse of bacterial growth. Now, you don't really get to dive in too deep with this bacteria, but you can see there is a lot of stuff happening inside this cell. And so much, there's literally thousands of chemical reactions happening at the same time. and it's actually able to make more of itself. You know, that's the fundamental quality of life. It can reproduce. Here, every bacterial cell that's being produced is essentially a clone, but what a remarkable process. Now, with a human, and I've gone through this type of procedure before, this is just IVF, here are embryos just starting to divide. Now, again, we kind of know what the end result of this is. It's a single cell that turns into something a lot more interesting. This is my daughter in Hawaii last year. I've learned if you bury kids, they can't run away. That started as a single cell. She started with a single cell. We all started with a single cell. This is remarkable. When we start to get good at programming DNA, you can make really complicated things, and they just happen. They just self-assemble. This is the most advanced form of manufacturing. This is biology 101 in a single slide. If you go into a cell and you rip out the chromosomes and you stretch out the chromosomes, you have about 6 feet of DNA, and that DNA essentially controls all the cellular processes. So we've been analyzing and learning a lot about DNA for a while, but here's the real take-home. Biology is an advanced nanotechnology, because all of this stuff works at the nanoscale, and it has a programming language. And that programming language is conserved from that bacterium I just showed you all the way to us. It really doesn't change. You learn one programming language, and you can program plant, animal, bacteria, virus. That's pretty cool. So we've been doing that. We've been learning how to analyze, read, sequence, and write code. And we have a whole new branch to the tree of life today that we call Synthetica. This is where humans have gone and changed the genome of an organism or created it from scratch. We have 3 tools, main tools, to really start to manipulate biology. One is evolution. The other one is editing, just making small changes. And the third one is actually design and synthesis. I'm just going to run through these quickly. Evolution, we've been using for thousands of years. Just in thousands of years, we've changed corn into the modern form from a very simple plant. Just in the last few decades, we've changed chickens because we selected for certain qualities of chickens that we think are really good for industrial processes, and so on and so on and so on. When Darwin wrote his book, he was actually trying to explain not artificial selection, which was in wide use at the time in plant and animal breeding, but how nature did evolution through survival of the fittest. We know that we can change the evolutionary course of organisms. More recently, we've been able to automate this process using a system called MAGE. It's essentially robotic accelerated evolution, which is pretty cool. But there's so many different ways we can still push evolution. Editing has really become popular in the last little while because of a new technology called CRISPR. I think the name is really good. It's certainly a lot better than clustered regularly interspaced short palindromic sequences. But CRISPR sounds accessible, and it is. It's a really low-cost way of doing copy and paste into virtually any genome. And it's only about 5 years old, but the amount of investment and work that's happening in CRISPR is remarkable. One of the world's record right now is doing 62 or 63 genetic edits in a pig to humanize the pig so that its organs are more suitable for human transplantation. That's pretty cool. We grow 1 billion pigs a year. If we can make their organs compatible with ours, we don't have any shortage of organs for transplantation. We've used CRISPR to attack cancer cells because it can do sequence-specific targeting of DNA. In other words, it can find the mutations in a cancer cell and selectively kill it. We can do it with antibiotics. By the way, this is a handprint of an 8-year-old child just on a Petri dish. That's just some of the stuff that's growing on their hand. You are more microbe than human. And we're already using CRISPR to do genetic surgery on embryos that we know are damaged from genetic screening. This technology has moved very quickly. And it's going to start moving into areas that are a little more playful. These are just glow-in-the-dark cats. I would buy one tomorrow. Like anything glow-in-the-dark, I would buy. I'm just that kind of guy. But we can also use it for actually de-extincting recently extinct creatures. And just a big shout out to Ryan and Stuart up here in the front, because this is work that they've kind of opened up and have been advancing. This is really breakthrough stuff. Nothing has to go extinct anymore if we have the code or we know how to recreate the code from something similar. CRISPR has also been used to make genetic edits in mice. This is about, a lot of people think that biotechnology is going to have you live forever. I'm not one of those guys, but this was one of the first experiments that really made me go, there's something here. And the mouse on the right here has one little switch put into its genome that basically says if any cell individually gets old in the animal, die. So all the old cells just die off naturally and are replaced with healthy cells. The one on the left just ages naturally. These mice are the same age. You can bet that we want to see this be tested in higher animals and perhaps people, because the idea of being 90 or 100 years old and actually being healthy is pretty cool. We're also learning how to hack intelligence. And there's not that much of a difference between the brains of a dog and the brains of a human, where our genomes are very similar. This might be your next Uber driver. Yeah, I don't know. Actually, I don't worry about dogs and this. Dogs are already pretty intelligent. It's when cats are your boss. That's when it's like, okay, game over. But the really, really interesting stuff, editing is cool. You don't have to write the whole genome, you just make targeted changes. But when you can start to write genomes, this is when it becomes really, really creative. And this is the area that I have spent most of my career on in the last 10 or 15 years. It's just, we now have printers that can print DNA, which means we can start to design DNA from scratch using electronic tools. Now, the amount of DNA you write correlates to the complexity of the program that you can create. So today, everyone has access to about 50,000 base pairs or less of DNA in terms of size of constructs. That'll allow you to do metabolic engineering and make single proteins, stuff that was really, it required a big biotechnology company or a state-of-the-art lab to do 15, 20 years ago. If you're really, really on the forward edge, you can do anywhere between 50,000 and 10 million base pairs. This allows you to do sophisticated metabolic engineering, make whole bacterial genomes, and start to work up to the first eukaryotic genomes like yeast. And this is still science fiction, being able to make whole large chromosomes and eventually anything larger. I put a baby there just to be provocative, but it could be a plant, a tree, a dog, it doesn't really matter. This is an example of metabolic engineering, one of my favorites. Back from 2005, it was some students that made bacteria work like photographic film. You shine a light on it, it exposes, and it can show an image. That was really cool. It got a lot of attention because, yeah, it's kind of harmless. It's kind of the biochemical version of what Kodak did a long time ago. Just this week, I noticed that they made it color film. These are bacteria that actually produce different colors. With light stimulus shined on it. And that's kind of cool. That's literally just a week old. Neat. That's just one example of metabolic engineering, but you can do a lot more important stuff like make drugs and do diagnostics, etc. This is just a frivolous example that you can appreciate. We're starting to do metabolic engineering to replace a lot of industrial processes. You've heard of biofuels. There's a whole bunch of other feedstocks that are a lot more valuable than fuels that are being produced by putting the code that makes it into cells like yeast and growing them in essentially big, giant industrial breweries. We're going to get some really, really interesting beers in the future. Trust me. Yay! But what I've been doing is whole genome engineering. I don't want to monkey around with just making small changes into a genome. I want to start writing them from scratch. And for doing that, you have to work bottom up because the technology is still pretty crude. So in 2014, with some friends, we made a synthetic virus that kills bacteria. It's essentially an antibiotic. So it's about 7,000 base pairs. man, I forget the exact number, but it's about, it's 5,600 base pairs of code. Wherever it boots up on this growth plate is where that synthetic genome booted up and started killing bacteria. So wherever you see a spot, it's essentially a successful experiment. It showed that our code worked, and it's actually making the virus that you see in the model on the left. That's really cool. But it was work that we recreated, but it's just that it was so easy to do. A software company could do it. That's kind of neat. Since then, we've actually gone to the very forefront of the field of making synthetic viruses. This is work that we did last year with a collaborator. This is designing a virus that actually kills cancer cells in dogs. The whole process is the same. You design the viral genome on computer. You synthesize the viral genome using some of these printers. You put the viral genome in the cells, which acts as factories to make the virus particles, and then you use the virus as a treatment for a dog cancer in this case. This is really cool, and this is what we're starting to commercialize now. I've spun this out, and I'm starting to build a whole company just to make viruses that give cancer cells the flu. If you have more resources and time and scientists, you can move beyond the virus and you can start making small genomes of bacteria. Currently, this is the only scientist in the world that's managed to achieve that. He's had about a 7-year head start over the rest of the world. He actually made a race car version of a bacterium. It's all stripped down, doesn't have anything that you don't need. There's no radio or air conditioning in this bacteria. It is essentially a minimal bacteria. About 500,000 base pairs of code. The thing that was really interesting to me was about a third of the genome. Even though we printed the whole genome, we still don't fully understand what that code does. So we have a lot of work to do still to understand the basics of living biology. But we're closing that gap really, really fast. At the very extreme of this work is yeast engineering. Yeast engineering has been moving along extremely quickly. It's all robotic now. They can make synthetic chromosomes. That is really cool. And now we're standing on the shoulders of the folks doing the yeast genome, and we've created a new project called the Genome Project Right. It's kind of the version, it's a new genome project for a new generation. 30 years ago, scientists made the Human Genome Project to read the human genome. Now we're aspiring to write large genomes like the human genome, which is pretty provocative today, but in 30 years looking back, I don't think so. I get to work with some amazing colleagues. George Church, one of the top genetic scientists in the country, recently voted to 100 most influential people. Jeff Buca, who is the leader of the Yeast Genome Project and developing most of this technology. And Nancy Kelly, who is just a force of nature in organizing scientists who are really like a bunch of squirrels on crack. We ran the meeting just a couple of weeks ago in New York. It was great. Hundreds of people from around the world came in, and all the working groups are established. So we're only 1 year into this genome project, but it's really getting some force, and down the road, really cool. And I just want to reiterate, we are not making synthetic babies. We are writing a human genome and other genomes, large genomes, and doing this work in cell culture. The really interesting stuff of making synthetic babies will fall to another generation. But, oh boy, it's going to come one day. This is, you may have seen this is a biobag. This is essentially an artificial womb and a little lamb that's been growing in it. Yeah, it's kind of Blade Runner-ish. Anyway, I'm going to just wrap it up and say biotechnology is changing super fast. It stands on the shoulders of electronic technologies, and it is really starting to take inspiration from Moore's Law. These are students that are essentially the masters of this technology. It's always the young people because they just absorb this stuff in the same way that 30 years ago they absorbed computers, or the current generation with social. The Bay Area is a hub for this because of its background in information technology. It's also got one of the best bio-incubators in the world right here in our backyard that has now incubated something like 60 different companies. It's going to touch every area of our life. I look at, the areas that this group is interested in. It's already touching agriculture because you don't need to grow a whole animal just to get a few products out of it like milk or egg white. So cellular agriculture is booming. It will absolutely change the way we deal with cancer and infectious disease because it's just changing the way the speed at which we can develop drugs. And yes, there's a dark side to this too. I wrote, as David said, a story about hacking the president and killing the president, but that was to sensitize the fact that we need better biosecurity and better biosurveillance. Because right now, biosurveillance is you getting sick and showing up at the hospital. You are the biosensor. That we don't have the tools for rapidly responding to pandemics, whether it's Ebola or Zika or the flu. We need this type of surveillance and we need this type of technology, and engineering biology is the technology that gives us that. It's also the only technology that I'm aware of that really gives us a hope at sustainability, because I don't think bulldozing another forest and putting up another iPhone factory is going to give us sustainability. It'll definitely keep us connected. It'll also allow us to start exploring beyond this planet, because we're not really good at doing sustainability in little small containers either. We know we can get to Mars. If we can keep living there is a completely different question. So we need a big investment in this technology. And finally, down the road, as we get better at this technology, yes, it really does change everything about evolution. Because this isn't just about selection. This isn't just about editing. This is about human intention being reflected on every living organism, including ourselves. And that means we all have a stake in this. We all have to be involved. Thank you.
Thank you so much, Andrew. My gosh. I mean, one of the reasons I invited Andrew here is he's one of the best explainers of this. You know, I have to deal with scientists who basically can't put two words together that we understand. And, you know, you can put together more.
I speak English.
2 words. You actually do. And I think there we go. We have this extraordinary rundown of really, you know, what's going to happen. Well, it's happening now, but slightly in the future. But let me, I'm going to ask you 2 or 3 questions, and then, you know, you all start thinking about, give you a chance to absorb that for a minute. By the way, how many people here have heard most of this? Okay, so we have a lot of newbies out here. All right, so you guys get ready for your questions, those who know a little bit about it, and especially those of you who don't. But Andrew, so this sounds amazing and it sounds wonderful, but what is real about it right now? I mean, you know, what's going to happen? How does this actually happen, both in terms of the science and technology, but possibly more importantly, society? You know, how do we absorb this?
Well, it's been happening for a long time. Like, biotechnology isn't new. Like, when I was a kid, I read about recombinant genetic engineering technologies in the early 70s. And you know, I've known people and companies that have really developed this technology for the last 30 or 40 years. None of this is new, just like computing isn't new. This isn't programming silicon, it's programming carbon. What's really happening is that the tools and technologies and the scientists and the researchers and the companies are now digitizing. And so that digitization accelerates. We've now got robots to do the manipulations. They work 24/7 and don't need a day off and can work with the smaller volume. So we're seeing changes in the speed and the economy and the accessibility that's just kind of throwing fuel on the fire. Last year, we had about $1.2, $1.3 billion invested in these technologies that we've been tracking. It's just been growing exponentially, and I don't expect it to stop for decades.
Well, in a way, it has been around for a while, and I've been covering this long enough. I mean, you hear these stories. It's a little bit of hurry up and then wait and hurry up. And I'm wondering, you know, we have people out here who are investors. We have others that are thinking about what is real about this. And part of this question is about society, because in some ways, it's not even the technology that might be hindering this, that might be keeping it from happening or happening in the right way. It's about society and, you know, the legal structure, the political structure. Talk a little bit about that, because you think a lot about this.
So there's definitely some headwinds, particularly in the U.S., because we have an established biotech industry, so there's a legacy industry that wants to protect their business. We have a lot of regulation, and we actually care about the environment and about people's health. So it's a little harder to get this work done. But when you push through those barriers, you have access to the richest society in the world, and you can often have a very successful company. What we're seeing now is that the tools and technologies are evolving so quickly, just as we saw with the internet, that regulation and education and really even just surveillance is kind of lagging behind the forward edge of the technology. And so people are just struggling to understand what it's about, what are the risks, where can I make money in investments, and there's always some hype to this. Everyone wants to get some attention to their work. So just filtering out what's hype and what's real is a big part of it. But like computers, we can pretty much guarantee that this isn't going away. Yeah. Like, life is pretty important. And again, it's got a universal programming language and a universal factory of the cell. So we can pretty much just expect that we're going to get more and more adept at doing this and find the right paths to market with things that people actually need and want. And we're working in a globalized world today, so it's not just US-centric. The technology is already distributed across the planet. So there's no assurance that the West will control this technology or lead it. In fact, a big part of getting the Genome Project right founded was the fact that we made the point that the technology was moving so fast. We're going to be doing large genome engineering in 10 years. If US scientists want a leadership role in it, they have to move now. And thankfully, they did.
Yeah, in fact, following the meeting, there were several hundred people. How many came from, countries other than the U.S.?
I don't have the exact demographics, but we had representatives from
everywhere.
Yeah, everywhere.
Yeah, China,
everyone that was working. China was a very big representation.
I mean, I had a couple of the other organizers actually in a panel last week in New York, and I had this realization as I was talking to them. We're going to talk about some of the scary stuff in a second, where this might lead, both good and bad. But it struck me that, first of all, this gathering was important scientifically to bring everybody together, because one of the reasons that these things happen more slowly than maybe they might is because science, it tends to be an individual pursuit, and people are working on it spread out everywhere. But
well, let me give you a little background. So Genome Science has been moving forward. Like, the Yeast Genome Project has been going on for 5 years, and it really involves some of the most amazing genetic scientists in the world. And the whole founding of the Genome Project was because in 2015 I was at the Yeast meeting, and everyone that was working on this was in one room, but there was most other scientists had no clue that it was happening. Here's some of the most advanced genetic engineering done in the world, and no one cares. There's no media in the room. And I just said, look, if you really want people to pay attention to the work, your next project should be to synthesize a human genome. And I don't mean, again, just do it in cell culture, the same stuff you're doing, but everyone will be interested. It'll drive engagement. It'll start all the discussions with ethics and intellectual property, and it'll finally engage humanity. But scientists are really, really conservative because their money comes from public funding. And if the public kind of goes, we don't want that, their funding just immediately dries up. Industry has a little more flexibility, but industry can't drive something so big and visionary that's 10 years out. It's just not part of the model. But if there's a pull, if everyone starts getting together, the discussion starts happening, and that's what has happened over the last year.
Well, it also struck me that there are some scary ways that this could go, and there are also some ways that it might not go. And getting this big group together, it's almost a firewall. You're creating an organization run mostly by scientists, and there's some echoes of that in the past, like if you all know about Asilomar and some earlier technologies where scientists got together and decided how to kind of ethically, philosophically handle this. And I found that struck me, talking to some of your co-organizers, that was a very important step to bring together the world, to kind of really make this out in the, and it was out in the open, and there's been a lot written about it.
Yeah, the first year it got branded in the media as a secret meeting, which was great because then everyone wanted to be a part of it. It wasn't secret; it was just under
marketing, huh?
Yeah, it was just marketing. But this was open and transparent, and I think we still have some ways to go there because we still haven't laid down the machine-readable version of the Human Genome Project or the Genome Project, right? Where all the electronic systems can really start start putting in data and start mining it. So I think there's going to be a whole new generation and a whole layer of electronic infrastructure that'll get put in place pretty quickly.
Yeah, let me just ask for a little bit about, I know you think about this and you mentioned it, but this is sort of scary. And for some of the people hearing this for the first time, you know, that we're on the cusp, perhaps, say, in a few years, decades of being able, and you talked about, you know, synthetic babies, but you said that is left for the next generation. But what do you worry about with this?
Well, so here's my motivation. I'm actually not too worried about most of this technology because biotechnology has been around for a while and we tend to be naturally conservative. But here's the thing, nature is always trying to kill you all the time. If you didn't have an immune system, you'd be dead in a few days. So every virus, every bacterium, every bug, every creature, even your dog would just eat you if you had no defenses. So I'm really sensitive to that. Right now, we're so blind to microorganisms, bacteria and viruses. We just don't have the tools to monitor them in real time. We have such a misunderstanding about cellular biology, the stuff that makes every living thing, that most people know more about their iPhones than about biology. That to me is scary. And the fact that we get so much, we're just not investing in the right areas to really bring together a community and build platforms. So that's kind of my fear. But I don't actually worry too much about engineered biology. As I wrote with my colleague Mark Goodman, this technology might be very, very good for making a targeted bioweapon in the same way that we make targeted bioweapons now to hunt down cancer cells or kill viruses. But I don't think I don't think it's going to be really good for making the next pandemic. If anything, these technologies will help us counter the next pandemic. And anything larger than a microbe, we don't really have to worry about because most larger things don't hurt humanity anymore.
Well, we can hope. And in fact, I think we're ready to open this up to you all with questions. And while you're kind of thinking about that, I think Peter's going to roam around a little bit, and we have a couple mics here.
So if people could just put their hands up if they have questions, and I'm going to literally run it around here as we go. And it looks like you got one right here. Why don't you go?
Yeah, this is actually a two-part question. Can you say who you are? Oh, I'm Julie Motz. I have a science and technology radio show on KWMR called Hot Tech, Cool Science. You know, when 3D printing got popular, I began to think, we've had the democratization of information, now we have the democratization of manufacture. And as I was listening to you talk, I thought, well, are we going to face the democratization of gene manufacture, and you know, where we can actually do it ourselves? And the related piece to that is, what do you think the ramifications of this are for changing the self? I think specifically of gender change and plastic surgery. But you know, what are the ramifications of that?
So I definitely believe that biotech is democratizing just as computers democratized. You know, where we are just entering an area of personal biotechnology, but we don't know how to do it yet. I remember buying my first computer in 1981 or something, and there wasn't very much I could do with it. And there's not very much you can do today as a neophyte in this technology, but the doors are opening. And when you get millions of new programmers coming into an area, and you're going to start seeing more acceleration and some interesting stuff come out, and some weird stuff. I usually say it's not faster, better, cheaper. It's also faster, better, cheaper, weirder when it comes to biology. As for what that means for re-engineering us, we're at the very, very top of the genetic pyramid in terms of complexity. So we're going to be the last ones to really receive the benefits of this. We're going to have to get a lot better at programming the single-celled organisms, programming model systems and organisms, and eventually we will start hacking ourselves. It's not too much to put in a single trait to correct a genetic deficiency. Putting in more traits is complicated. We don't have all the technologies for that. But I think we're going to keep opening those doors up. So I know how we came into this century. I don't know how we're going to exit. But I don't necessarily think that taking control over the reins of evolution with our intentions is a bad thing. I don't think it leads to the master race. I think it just leads to kind of mass personalization and some really bizarre changes. But we might need to engineer ourselves if we want to go live in space or live on Mars, or really just reach our full potential. So I'm open to those ideas, but I'm a little more liberal, and I have a higher risk tolerance because I'm old. I'm going to die in 30 years. So.
I know, we may just miss this, Andrew.
I'm not too worried about it. It's called cloning.
Okay.
Thanks.
Preserve,
Yeah, sure.
preserve yourself. And by the way, people ask about the safety issues here, too. You know, some of what he said may or may not be right. I mean, we've seen some of these things play out in sci-fi, you know, clone armies, things like that. So, you know, push Andrew a little bit on this. Yes?
So I'm Mark Bregman. I'm a physicist and CTO in a tech company. And I was one of those young people who got involved in silicon. So my question is that as I saw the evolution of VLSI, there were really three things that evolved together: the tools, the processing, the printing as you might call it, but also the architecture, what we wanted to build. And a lot of what you've talked about is using the tools and the printing technology to replicate architectures we already know about.
Right, and so VLSI is very large-scale integrated computing, essentially chips. Yeah, we're not really good at biological design yet. Most of the first-generation biotech industry just made single proteins. We still don't fully understand how proteins fold. Today we're doing metabolic engineering, but it's still pretty much see-spot run up a hill. Not that sophisticated, but that's part of the reason why I'm with Autodesk, because we build software tools that do really, really complicated things and make that work pretty easy, like modeling and simulation and fluid dynamics and others. So I have hope that we're going to be able to design really complex things with very powerful software tools that have built-in regulatory, built-in oversight, and a lot of intelligence that, you know, to prevent the easy errors. You're probably familiar with a company called Mentor Graphics. One of the first groups that kind of made these tools for designing chips. I was really fortunate that earlier this year I got to go and present to them, and it just feels like we closed the loop on a full cycle of VLSI engineering.
Yeah, this is another reason why getting these scientists together. These are, you know, mostly careful people, and we do need to design, you know, how to keep this safe along the way. And, you know, kudos really to you guys. It may have been a little secret before, but having it open is really, really important. And, you know, again, Andrew's talked, there's so many things to talk about with Andrew. He's actually called for full transparency on this, including the president's genome in his article. You know, that's maybe one way you deal with this, which is an interesting sort of philosophy here in the Valley.
Well, we actually predicted that high, you know, noteworthy individuals would have their genomes kind of published by biopaparazzi, because it's so easy to get genomic data. And it's only about $1,000 now to go and get your full genome sequenced and analyzed. It's just not as readily accessible as you know, as just sending off a photograph to a publisher. But it's going to get there in the next few years. So.
In fact, when Barack Obama was running for president, and I met him, and I shook his hand and said, you know, hello, and his water glass was sitting right there, and I was thinking, wow, it'd be an interesting article to just stick that in my coat, and maybe get John McCain's and then do a comparison. And that was way back, you know, 2008. Anyway, other questions? Yeah.
Hi, I'm Raphael Latterman. I'm a general entrepreneur with some biotech background, previous biotech company. I've been concerned for the last 30 or so years that what will happen is that people will be able to do what you did in a laboratory and create a virus that will just be able to kill everyone, and they'll be able to do it in their kitchen. And so one concern I've had is that that's not compatible with a free and open society if you might have to control that. So one possibility is that the countermeasures will be equally easy to produce, but what would be your take on that?
Well, my general take on it is, look, nature is actually trying to make those viruses at a giant scale in every ocean, every air, every organism, and trying to kill you, and you're still here. There are ways that you could use this technology to build something that is engineered to kill a large number of people, but we have very few examples of that actually successful. The one, when I was a grad student, they still hold up all the time, is the 1918 influenza virus. Because we had been fighting World War I, and killed 8 to 9 million people in the trenches in Europe. And from that trench warfare, we evolved a particular strain of flu that when the war ended and everyone went home, they seeded around the world. And flu doesn't kill you. But in this case, it was such a virulent flu, your immune response killed you. And it killed somewhere between 50 and 100 million people. That is really the only recent example, I mean in the last 100 years, of something that wiped out a single-digit percentage of humanity, and it was a follow-on product of our war. I think we have a very narrow window where this technology could be co-opted to make a pandemic, but because it can also be used to make an artificial immune system and defense system, I think that window is closing extremely fast. And because it's built on electronic technologies, the robots will survey all the digital code. The stuff you have to worry about today, in my opinion, is electronic code, because that's what runs the world. It runs your car, it runs the airplanes, it runs our banking system, it runs our medical system. You just saw the hacks of the National Health Service in Europe. Electronic code is a lot more brittle. Electronic processors are a lot more brittle than anything biology has ever produced. So I recognize the risk, which was why we wrote the article for Hacking the Presidents. We want to stimulate next generation biosurveillance and biosecurity and biodefense, because the current architectures are just way too creaky and can't do it. But it's not my big global fear. My big global fear is just heart disease and cancer. That's what's probably going to kill me, or some microbe that my doctor can't identify and doesn't have a treatment for.
But this technology, Andrew, is
naturally produced.
This is relatively simple. I mean, a high school student 2 years ago, in fact, in Georgia.
It's not. Yeah. It's not that simple. I know it's not that simple because I just made the world's largest and first therapeutic synthetic virus with my colleagues, and it was like pulling teeth. It was hard, it took a long time, it was expensive, it took me pulling together some of the best people in the world, and it is,
I mean, I want to be clear, doing a synthetic organism is tough, but
it's really
say
hard.
Gene editing has, with the advent of CRISPR, has become cheaper, easier. There's a high school student that won the Intel Award a couple of years ago, and so this is not, we always compare to splitting the atom. It's actually really hard to make it's easy to make a nuclear bomb, it's hard to make
it's pretty easy to write computer code that infects computers too. So all of these technologies are dual use. So it's always about the intention around the creation and deployment of the technology. And right now, we're just not that sophisticated when it comes to biotech. There's much more risk with computers and robots, and even social engineering. So yes, we will always have risk. There will never be zero risk in any technology. But I think in this case, because we are made of the stuff that we're learning how to manipulate, and as is much of the world, and we depend on it every day for our existence, just food, for example, let alone medicine, I think the benefits in general far outweigh the risk. But we have a natural, built-in, and in many ways, societally reinforced fear of monkeying with biology, and I accept that. So that's why we all have a stake in this game.
Which is probably a good check. Yes.
So folks who want, put your hands up if you have questions. I'm actually roaming from the back here too. A lot of questions, and okay, see some over there. But right now it's going to Sunil. Just introduce yourself.
Hi, I'm Sunil Paul. I'm a serial entrepreneur investor. So as I'm listening to all of this, I would love to get a reaction to, and you're kind of touching on it now, but it feels like every time we come upon these new capabilities, our imaginations take us to a place that is kind of rapid projection of what's already happened. And if we kind of compare what happened with sequencing the genome, all kinds of projections of curing cancer and all kinds of wonderfulness that were going to happen if only we could sequence the genome, and then it turned out, oh, it's actually way, 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 way more complicated. And now all of the discovery seems to be around how complicated, how hard it is to do anything meaningful and helpful. So, I don't know, make the case that it's not as hard as that, or maybe it is really that hard.
So what I'm going to say here is, if I'm talking to an investor, It's easy, it's wonderful, and we'll get it all done in a couple of years for a really low amount. Just give us the money.
Yeah. Sign here.
If we're looking at the darker side, we talk to Hollywood, and I know because we optioned the hacking the president's DNA story to Hollywood, and they keep re-upping it. It hasn't been made into a movie yet, but you never know. But they keep re-optioning it. So we explore the good and the dark by investment and storytelling. It's all storytelling at the end, I guess.
It is pretty fascinating. I mean, that's why I was asking earlier about how society reacts. But there are lots of questions out there. Yes.
Oh, hey, Andrew. Omri. So I'm from Twist Bioscience, one of the actually making DNA here in San Francisco, one of the DNA printers. And I want to take it back to designer babies, because I'm going to have a baby girl next month after 2 boys. So I'm very happy about that. But if I could edit her to make her live longer, to be smarter, to be healthier, I would do it, but the knee-jerk reaction is to ban all this technology, and I'm not allowed to do it in the US. Can you comment on the reaction, on the knee-jerk reaction not to do it, to ban it, and not to pursue the positive things you can do with it?
Well, first, congratulations, and two, I understand, because I was never going to have babies. I was just going to have clones and bacteria. And then I met my now wife at a TED conference, and I knew that I had to marry her because I wanted to have babies with her. So that took some surgery, and then I ended up having to use IVF. And so I just said, give me all the latest technology you've got, because I really wanted to understand the latest technology around reproduction. And our baby, she's approaching 2.5. She was booted up in New York, in a New York lab, and it was super low tech, super low tech. We just did another one a few months ago here in the Bay Area, and it was much higher tech. They had all the genetic screening, they had robots, they had full lab tours, I'm happy to say that the genetic technologies we have today can find really significant errors in an embryo and diagnose what the problem is. We do not have the experience with editing technology to repair those embryos yet. And that's the very forward edge of the research, genetic surgery on embryos. But there is such an important driver for learning this and applying this to parents will ask for it. I have nothing against it. What other person had any right to kind of dictate what I was going to do, to help my daughter have the best life she could? It's part of our challenge of parents to give our kids the absolute highest step upward that we can do. So I have no problem with it. Different regions, different people will have some issues, but this will all sort out because it's a big world and this technology is global and rules and regulations are not universal, and our experience will grow with this technology. So, I think it's really fascinating, personally.
I'm sort of curious, how many people, if it was legal and safe, would genetically engineer your child to be smarter, stronger? How many people? Okay.
I just want glow-in-the-dark eyes.
How many people want to be glow in the dark?
Well, I don't want to glow in the dark, but I want
off.
night vision. Like, I would love laser eyes and night vision. The intelligence thing, I'm not convinced is all that useful.
We don't really know even what it is.
Yeah, but I would like to be able to eat Doritos and drink all the fatty stuff. And I don't know.
Okay, how many people?
Look, one day there'll be an app store. You'll just be able to choose some really awesome traits, but I don't think we'll all choose the same traits, just like we don't download the same software or buy the same cars.
I think we had probably about a third that would, so maybe 2/3 that wouldn't. So, other questions? Yeah. Yeah, tell us who you are, too, if you don't mind. Yeah.
I'm Fawn Skiles. I work at Buy Right Market. I'm in the food industry. And one of the concerns that we face with commodity food is that the overuse of antibiotics. And I've gone to conferences where they're worried about basically not being able to have antibiotics to work against the superbugs that are coming up. So how would this disrupt, the future of our livestock and our food production.
So it's important to kind of understand antibiotics and just evolutionary pressure. Bacteria grow so quickly, you can get antibiotic resistance generated in the lab in just a few days. I was going to play a video that was a really great little video from Harvard where they show that you can take standard E. coli and make it absolutely resistant to an antibiotic in 11 days. It'll use the antibiotic as a food source in 11 days of growth. Then there's the whole business of making antibiotics. Penicillin was the first one. It took 10 years to get penicillin, convince scientists that it was useful, and then start to make it at scale. It only really started to appear in the Second World War, and it saved lots of lives because people were doing all sorts of things that damaged themselves and caused sepsis. But we kind of hit peak antibiotic in the 1980s, and where we had a whole library of them. We said, microbials, we've conquered the microbes, and we stopped making them. And here's the problem for the drug industry. When you make an antibiotic, you give one treatment to an otherwise healthy person, and they're cured, and they don't come back and buy more and more antibiotics. So it's not a business that most of the pharma companies want to get into. It is absolutely easy to make antibiotics to any microbe. It is absolutely easy because every microbe has a relationship at least with one virus. You can use CRISPR constructs to selectively target microbes. You can build all sorts of engineered nanoparticles, and you can start making all sorts of new drug combinations too, or finding them in nature. The problem with pulling stuff from nature that might be very complicated drugs is we don't know how our bodies are going to deal with them. But we do know how many of these engineered particles will work because they're made of the same stuff that we are. So there is no reason why we have to run out of antibiotics. If anything, they're going to become more targeted and more selective to the particular microbe or virus that you've got. So I think we'll see a next wave of antimicrobials, and we'll start to use much better evolutionary design so that we can minimize resistance in our agricultural, food chains.
Yeah, the microbiome is really beginning to take off too.
We're just starting to map
yeah.
it. Like, remember, we mapped the human genome before we mapped our microbes.
And yeah, I mean, you follow all of these stories, as Andrew said. The 20 or 15 or 20 years I've been covering it, these stories have been around the whole time. And they move, fits and starts, and it's interesting to see when they sort of break out. And synthetic biology is doing it right now. The microbiome, it'll step back.
It'll
again.
actually hit the virome one day.
The virome.
Yeah,
yes.
because there's just as many viruses that infect us and cause disease, but we don't even talk about it because we don't have the right assays for it. You can't see a virus under a microscope. You can see a bacterium. You need an electron microscope to see a virus. So the actual best microscope that we have today is genetic sequence, because that's how we can actually look for any living creature.
And the antivirome. Other questions? Who's got, yes.
Hey, I'm Chris McGinnis. I'm a grad student at UCSF. My question relates to the distinction between a functional synthetic genome and a synthetic genome. So when you make a genome, you can have like you talked about, it's 6 feet of DNA, but for it to form a functional organization, you need the products of that genome to form the chromatin structure that makes sense in a given cell. So it seems like there's a bit of a chicken and an egg problem, and like what is the major barriers before we can actually sort of create life from the ground up?
Yeah, so we're not building life from the ground up. We don't actually profess that. What we're actually doing is writing synthetic genomes, and that's like coding an operating system, but we're not building the computer. The computer is the cell. So we're just loading a new operating system or app into that cellular environment. Now, it's not that much work when you can start writing whole genomes and working with cell-free systems that you couldn't boot up a completely synthetic protocell, kind of a minimal version, and building all of the biochemical components from scratch. But there's no real reason to do it because every life form from bacteria up to us uses a conserved set of cellular machinery. And if we can just learn how to program that machinery, all of the diversity of biology and more, all the previous versions of biology that happened before us, including dinosaurs, and all the biology that wasn't selected for by natural evolution becomes suddenly open. It's a giant sandbox. even if you don't change any of the machinery or the coding, the standard genetic language. But just being able to write genetic code faster and cheaper opens up the creativity of how you're going to reprogram cellular life.
But you are talking about, I mean, maybe not you specifically, but some of the synthetic biologists at GP Wright about these super cells. I mean, cells that would.
Well, we're just reprogramming the genome so that we're essentially modifying some of the subcellular architecture as well as the genetic code so that it's resistant to every known virus or other infection, and that we can minimize cancer appearance because we boost some of the cellular machinery that does error checking. We change the genetic code so that it's resistant to viruses. Yeah, so that's just because we use cells as manufacturing plants for things like medicines. And if a manufacturing plant gets infected with a virus, it can shut down the whole manufacturing line and the entire thing has to be sterilized. So if we can make cells that are resistant to damage and to infection, then it's really good for industrial biotechnology and drug making, for example.
Yeah. Over here.
Hi, Michael McElligott from the Long Now Foundation. So you said lots of things are changing, there's lots of innovation going on. Can you characterize a little bit more what the ecosystem's like right now for that innovation and what the stopgaps are, where the innovation's happening? I'm curious, in high tech right now, if you gave them more programmers, tons of things would happen. It's at that level, but is it hardware dependent? Is it people coming straight out of school that are doing these startups? Is it veterans of the industry? What's that kind of look like, and what needs to be broken down for it to go faster?
No, that's a great question. So the ecosystem is definitely be changing. Most of the people that we hold up as like the top genetic scientists are all like me, gray and old and kind of crotchety. The whole new generation is like the iGEM photo. They're all kind of under 25. They're all hot on this stuff. They get it. They've grown up digital. You don't have to teach them exponential technologies. They'll work for next to nothing if they can just get do something interesting. Most of them are still living with their parents. It's remarkable. So the ecosystem is definitely changing. It's getting younger, more digital, faster. There's lots of money flowing into it. People still don't know exactly what's going to be the breakout. We haven't got the Google, the Facebook, the Apple of biotechnology and next generation biotech, but boy, we're on the cusp. I think it has a chance of happening here because the US actually has trained some of the best scientists, has great investment architecture, some of the best technology. For me, the bottleneck, we still don't know how to design well because scientists haven't even come together to make a virtual cell model yet. We still have no Google Earth, for example, of the cell that we can all collectively add data to and manipulate. So that's something that we'll see appear in the next few years. And for me, the biggest bottleneck is the DNA synthesizer. Because I don't want to work on the DNA, natural DNA molecule that I'm pulling out. I want to be able to write that code from scratch and verify that the code I wrote is the code that printed, or the code I designed. So the DNA synthesis technology is really, really crap today. But we're going to see, just as we saw with DNA sequencing, an incredible leap of synthesis capability in the next few years. That's what the Genome Project Wright is pulling. It's pulling by saying that we are going to now start designing and building large genomes. That's what we want to do. It pulls all the technology forward. So we're going to see a big shift there. And after that, it's really about just making the kick-ass killer app, no pun intended, that people want and will go and search and buy, even if it's a gray market, and it'll just suddenly revolutionize the industry. But it's going to happen with the young, for people that have a need. They're either sick or they're hungry or they just want to see some broken system get better, and just the right place at the right time to spark the fire. There's a lot of fuel on the ground. We still need a lot more, but there's definitely a lot more sparks starting to hit that fuel.
Is much of this ready to be industrialized? I mean,
at scale, no.
Yeah.
No.
Okay.
The very first examples of doing this stuff, not with people, but with robots, have kind of appeared here in the Bay Area with Emerald Labs. It's a robotic lab. Zymergen, Ginkgo on the East Coast. Just Transcriptic. These are the first these are the first facilities that are really starting to do high throughput industrial biotechnology with the next generation tools and technologies. Twist is doing high throughput industrial DNA synthesis. But this really is still hard. It's still work. And it isn't until this architecture becomes just easier to access, is more digitally connected. It probably still drops an order of magnitude or more in cost, and the time to do the work drops by an order of magnitude, that we're really not going to have the breakout super exponential acceleration. But it's guaranteed to come.
Well, it obviously took decades to create
to get to this point.
Google.
Well, it took decades,
in
to
IT,
build the IT infrastructure.
Yeah, and we're probably just at the very early stages of ARPANET, or even before.
But I'm always looking for that. Like, I'm always looking for those dots that just stand up above the background, because I can see where this is going, and it's exciting. I just don't know who's going to lead. I don't know when it's going to start exactly or how quickly it's going to grow. But death taxes synthetic biology. I know it's just going to happen.
So I have a question. Yeah, over here. Elizabeth Carney, I'm an entrepreneur and health professional. And my question is about that complexity part. Because I think we've been talking in a very mechanistic model. It's very much zeros and ones. And yet in nature, what we find is something that's much more holistic. And how does that inform us? So that's the first part of my question. And the second is all that stuff that we used to call junk DNA that we discovered was actually the epigenetics, like all the lifestyle intelligence that actually informs whether your grandfather's heart disease gets you or not. How is that taken into account in this new world?
Okay, so the cellular processor, the cell, is definitely squishy, and a lot of things are happening in real time, and it doesn't have the same rigid architecture as computers. But the code that drives all that is still digital. So it's not ones and zeros, it's A's, T's, G's, and C's. But you can make direct correlations between the function of the squishy processor, even though it's complex, and the digital code that makes it. It's too many variables for a human mind, but it's easily accomplished by software. So software tools will become the primary drivers for design of cellular metabolism, and it will work bottom up. Craig Venter's work published last year with the synthetic stripped-down bacterium is a really great example of this, because we already know that if we pull out any code from that organism, it dies. This is all essential code, and we're closing the gap to understanding what that code drives in the cellular metabolism. So we will have a very good, reasonably accurate digital model of a cell. But for a human to look at it, they won't understand what's going on any more than you could look at all of the electrons flying around a chip today and understand what's happening. As for the kind of the higher order features, let's just say we've got a lot of room to get there. We've got a lot more stuff to understand, but it's all tractable. It's just going to take a while to deconvolute the complexity.
Well, part of it is we didn't build this.
We didn't build it, yeah.
Like 3.5 billion years of evolution,
it is essentially an alien computer system that we're trying to reverse engineer as well as program so we can do something useful with it.
And by the way, it's worth saying that this consortium that got together, these efforts, they're trying to, at base, understand what is going on. In the Minimal Genome Project, for instance, as you said, this tiny little organism that Craig Venter created, a third of it we still don't know.
Yeah, but it's only a third.
Like
it's like 149,
I guess that's better
147 genes. Like, it's basically 1 year of PhD projects. You could farm out the unknown code to groups of people, and it would be solved in a short while. And I was just recently contacted by a group that was building a new fully digital whole cell model. So I do actually believe that we will have digital versions of cells that, kids will just strap on VR gear and go flying around on their favorite molecule and understand what it does. if you haven't tried VR and how immersive it is, it's not really a joke.
You may have cells driving it too.
Yeah.
I mean, these can work as computers.
But we definitely need this computing architecture and unbiased computing architecture. I think we need things like AI and deep learning and machine learning to deconvolute some of the complexity here. Otherwise, we are going to put our own biases on some of the data that comes out, which we've seen time and time again in the R&D fields. It might get your paper published, but it's not reproducible. So this has to be
Time for a couple more questions?
Yeah, we're just getting to towards the end here. And so last few questions. Actually, I was just curious if Stuart and Ryan, because you're in this space, is anything that struck you that you want to ask a question or comment on? Here, let me give it. Oh, here, you could. Okay, because you're going to answer.
Yeah.
Swap you. Ryan Phelan with Revive and Restore, working on endangered and extinct species. And one of the greatest new advances following on the success of CRISPR is with a technology called gene drives. And I was sort of surprised you didn't mention it because it has gotten a lot of controversy. In conservation, we're looking at gene drives as an opportunity to remove invasive species that could have a tremendously great impact for endangered species. So can you say something about that?
Yeah, so gene drives is a way to hijack evolution to produce a certain outcome. And it's getting most of its attention right now because it's being deployed to essentially cause mass sterilization of mosquitoes to reduce mosquito populations. But you can develop gene drives to advance and propagate any genetic trait. So it's kind of a positive feedback loop for genetic selection. I didn't mention it because I'm still on the fence about it, and because put this way, I'm even though I'm not into killing any organism or driving any organism extinct, and I hate mosquitoes, and I understand how much human suffering mosquitoes cause, and it's not just because you get some bites; it's because it's a major vector for disease. But I would much rather engineer mosquitoes so they don't bite humans, because I don't want to disrupt all the food chain, which we don't understand yet around mosquitoes and other life forms. So right now, gene drives are new. They're as new as CRISPR. We're still exploring them. There's certainly some positive examples, but you will certainly see a movie or two about the negative aspects of gene drives too. But it's one of these very powerful new technologies that is already being deployed in the world. But it really comes under the first heading I had about driving evolution.
Okay, a couple more, yeah.
Oop. I think, okay, we'll get one more over here, but then if you want to wrap, and then we'll get one. I saw the hand over here. Go ahead.
Thanks. Lorenzo Trefethen. I'm the creative director at Trefethen Family Vineyards up in Napa, and very interested to hear about yeast. But you've kind of been talking around it. There seems to be something very different about this technology than all the other technologies up on the little graphic. And that is, when they were getting started, I don't think anyone really cared about what silicon was doing, just out in the general population. This one does have the potential to directly affect us in profound ways. And so I think that there is, at this early stage, much more attention here. And it seems to me, I know you're very optimistic and think nature's trying to kill us, so it's going to be hard for us to do better. But doesn't it kind of hinge on that first really useful or really destructive application? Like if someone creates the next Google, great, but if someone reboots smallpox, is it really unstoppable?
Great point, but I look at some of this. So biotech is the only thing that connects all of this in some ways. But every single one of these technologies probably has a group that is going, hey, should we do this? Robotics, we used to think of killer robots. All of the AI stuff, I remember watching the movie War Games a long time ago. You know, and biotech is just one more technology. Probably the reason why we're a little more fearful of it is it's the most powerful technology out there. I like, you know, it really is. Like, it is the most powerful technology we know about in terms of manufacturing and computation. But we didn't make it. And we don't fully understand it yet. So we are, and it does get tied up with all sorts of creation stories, religion, and even our own sense of who and what we are. That being said, it works really, really reliably, and it has amazing defensive systems. I'm always amazed. I've been building a company and building a baby. You build a baby, it just happens. You know, like it just happens. Companies, nothing happens unless you go do it. But you feel like you have some control over the process. So I think that's part of the dichotomy, and the only way we'll get more comfortable with that is just by doing it. So you know, that's the way I look at it. But it is really the only technology that connects all of us. And I can make the case that any single one of these other technologies is just as scary, or there's people that think about the scary side, because I've seen movies on just about every single area.
Yeah.
Yeah.
I guess we should probably start wrapping up. Peter, do you have any closing?
Ooh. Yeah, I think, no, in terms of questions, no. I have totally entrusted you. I think you've done a fantastic job. I think we should give both David and, of course, Andrew a fantastic welcome. And as is always the case, there's still food and drinks here, and the whole idea here is there's a lot of folks who would be great to kind of connect with here, and also to connect here with both David and Andrew. But thanks so much for everyone coming, and stay around, schmooze a little bit. And next one we're going to have, June 20th, is basically going to be around autonomous vehicles and a lot of the future transportation stuff. So always a great time, but this was a fantastic evening. Thank you so much.
Thank you all, too.
Thank you.